Yeah, that should be good, right?
Okay, so in this section we will go over about chemistry knowledge. So the reason is when we later on, if we talk about like meat tenderization, they're gonna talk about glycolysis a little bit. So I want you to just have an idea what is that and pretty much overview all the basic biochemical knowledge in this like 1 hour time. So first of all, this is easy. When we talk about metabolism, we have separated them onto two, which is anabolism and catabolism. Okay, and if you want to look at here, catabolism, you generate ATPs, and this is we use ATPs and the biosynthesis. Okay, biosynthesis. So that's totally opposite direction, and it combines them. We call it metabolism. And you can see here, you will see the energy resource source usually is glucose or sugar, and all the way become a waste products like acid, carbon dioxide. And in the other ways, they take the nutrition, they form the small molecules, they become a large molecule, then macromolecule, and finally the cell wall structure. So when you see here, you can see the slides, totally different two process. And most of the time here, we talk about these catabolism. And anabolism, we actually will talk separately when we talk about the lipids, protein, and amino acids later on. Now, this is a metabolic pathway. Metabolic pathway, we have basically have those like three. So number one is usually we say it's linear. So let's say A products go to B, go to C. Number two is quite branch. Branch, you have A goes B and C, then you go D and you go E, like that. Number three, we call it circular, which means it's a circle. Okay? Now, you know this. If I want to give you an example, what is linear? Fermentation is a linear metabolic process. Branch, glycolysis. Glycolysis. How about the last one? Citric acid cycle. Or we say TCA cycle. So those are the examples of those three types of the metabolic pathway. And you know about these, just very briefly mentioned. Now, we need something happens for the biochemistry reaction happen really quickly. So what we need? So we have a substrate. Okay, substrate, this is end products. This is most of the time is sugar. And the end products could be acid or carbon dioxide. What we need, we want this to happen real quick, we need an enzyme. Now enzyme basically is a protein. And we need to know the enzyme is not involved in the biochemistry reaction. It's not involved in the final products. It only let the reaction happen more rapidly, and we say it's more efficiently, because the enzyme is decreased the energy required for reaction. And in the food chemistry class, we have a certain chapter to talk about enzymes. If we have time, then we can get on that part. And you can see here, if you see the energy is right there, this is what we need. If we do not have enzyme, when we have enzyme, we can decrease to that red parts. So basically, if this is the energy what we need, This is no enzyme. And this is the one we use enzyme. Okay, so these are the basic stuff. Now we talk about the ATP. Remember ATP is the energy currency, ATP we talk about. Adenosine triphosphates. And how are we going to get it? Basically, you have three methods you're going to get it. Number one is substrate level phosphorylation. Number two is oxidative level phosphorylation. Number three is called photophosphorylation or photosynthetic phosphorylation. Now, we don't talk about this guy here. This is usually we talk about this plant science. We major talk about here is substrate level phosphorylation and oxidative level phosphorylation. Now, what's the difference between them? If we want to say very easily to say, substrate level phosphorylation, which is now involved in the electron transport chain, and this is will be with electron transport chain and generate protein motive force, PMF, for oxidative phosphorylation. So this is basically what happens. And we will give you some of the examples. Okay, this is something talk about electron transport chain. So when everything talk about here is an electron transfer, carry on the energy. So this is an electron. They always transfer from a donor to an acceptor. Okay, how do we know it is a donor? How do we know it is an acceptor? So for example, we know NAD plus and is a good donor, the electron donor, and an electron acceptor is oxygen. Is a good electron acceptor. So for example, compared to oxygen, we also know nitrogen is a good electron donor and oxygen is a good electron acceptor. So how do we know about that? It's based on the concept of what we talk about called electron negativity. So the electron negativity value is higher, which means it's more pretend to be an electron acceptor. If it's lower, that's more pretend to be an electron donor. And in the experimental parts, they did do a two-half reaction. Two-half reaction, they usually to testing the voltage value. So the voltage value is more negative, is more pretended to be an electron donor, and more positive tends to be an electron acceptor. So there is a two way to explain that. One is electron negativity, second one is they're doing a two-half reaction to see the voltage value of those chemicals. Okay, so this is showed you here, which is always move from low affinity molecular to high affinity molecular. What means a high affinity molecular? Which actually means they have a more electron negativity value. Okay, so that's what happens. Now this is the one, another thing. So we have a two electron transport chain. Okay, so I'll just draw one example. This is the one commonly happens is NADH, FAD, FMN, FES, then go to COQ, coenzyme Q, then go to cytochroma C, then go to cytochroma A, and then go to cytochroma A3, and then go to oxygen. The electron is transferred like this. Okay, now, here is a question. We will know if the electron transfer starts from NADH and the final is oxygen, they will generate the amount of the energy is higher or greater than the electron started from FAD to oxygen. Why? Because the voltage value difference from here to here is greater than from FAD to oxygen. This is a voltage value. The difference is greater from NAD to oxygen, and the greater from NAD to oxygen than FAD to oxygen. That's why this is all the difference. This is the difference of energy. Okay, this is what this sentence means. More energy released when difference in electron negativity is greater. The greater of the electron negativity, which means the correction, is the greater the difference of the chemical to accept the electron. That's why more energy will be released. Okay? Now, here is something else, and actually a very basic knowledge. I hope everybody will be understanding. We review a little bit of chemistry. This is called oxidation, reduction, reaction. Now, most of the chemical reaction or biochemistry reaction in the bacteria, animals, or in the metabolic pathway, or even in the food products, cell oxidation, reduction, reaction. Okay, therefore we need to know some basic stuff. A and B. And also most of them is reversible, generally C and B. Okay, so here is a question. If the electron is transferred from A and B, A to B, okay, let's say electron transfer from A to B. Now you're going to tell me which one is oxidized products. Which one is a reduced products? Which one is an oxidizing reagent? And which one is a reducing reagent? Okay? And this is generated E, let's say E accepted here. Okay, now you tell me, which one is an oxidized product? Which one is a reduced product? Now here, when the A go to B, generate C and D accept electron. So how to understand this? There is a term we can use it for. Usually we go to graduate school, we need taking an exam called the GRE exam. I know WVU is not required in our division. Why you usually say this term? This is easier to remember. Reduction, which means you gain electron. And the oxidation, in other words, is you lose electron. Okay, so here, the D, chemical D, get electron. What that means? That's a reduced product. Therefore, in other words, chemical C is oxidized, they lose electron. That's why it's C. Okay? Now, chemical from A to B, which means A is oxidized. So A is a reducing product. And B, A is an oxidizing reagent. B is a reducing reagent. Okay? Sorry. Should be like this. Reducing reagents, they supply the electron. This is supply electron. And oxidizing reagent, this is accept electron. Okay, so that's what happened here on the slides. This is all reduced, oxidized, so you can see here, X lose electron, X is reducing agent, Y gain electron, Y is oxidizing agent, reagent. And the C is oxidized products because it's a loose electron, and the D is reducing products because it's gaining electron. Okay? So that's what happens right here. Now, this is slides tells you they basically have some of the electron carrier during the electron transport chain or during the metabolic pathway. What are they? Typically, which is NAD, is NAD, NADH, FAD, FAD, H2, and NADPH. You are looking to, okay? NADPH. And now, we don't talk much about here. This guy is used in the Calvin cycle or used in the pentose phosphate. Pentose phosphate is an electron, is a metabolic pathway related to generate DNA and RNA. We don't talk about this too much. In the metabolic pathway, we major talk about NAD, NADH, and FAD, and FADH2. These we use a lot. Now, how many ATPs they generate during the reduce from NAD plus to NADH? We usually say it generates 3 ATPs. How many is it reduced from FAD to FADH2? We say it's 2 ATPs. Now, this is the old rule or old policy. Right now, they take consideration of the ratio between oxygen and phosphate. So they think NADH reduced to NAD reduced to NADH2 is 2.5 ATPs. And the FAD reduced to FADH2 is 1.5 ATPs. Now in this class, when we do the calculation, we still relate it on here. We still use this formula, okay? We still use this number, we don't use that number. That's, lots of the new textbooks, they use it, but we still use this. Now the electron carrier is very important to carry on the electron transfer. And there's a bottom line is, for the live body, for example, let's say for bacteria, these electron carrier they must be recycled. Otherwise, certain live body will be dying. And I will show you an example when we go to talk about glycolysis. Okay? So bottom line is this electron carrier, especially NAD plus and FAD plus. These has a well which is available to accept another electron. These has to be recycled. If they are not available to be recycled, which means the bacteria or the live body will be dying. So from this perspective, why the human being or animal will be dying? At the end of the day, they are completely oxidized. There is no recycled NAD plus to be carried on or accept more electron. Okay. Now here they listed something right here. I'm not going to go over this detail by detail. It just tells you the function of the NAD, FAD, and NAD pH. This usually used for the biosynthesis. And these generate the proton molecule force and generate the ATP synthesis, triggers the ATP synthesis. And these slides I will just go over real quick, which means during the metabolic pathway, we will generate lots of the products. Those we say it's a precursor. So they all contribute for the next components generated during the metabolic pathway. But they will be like a transition one sometimes. So you see, glucose 6-phosphate coming from glycolysis contribute to lipopolysaccharides. Pyruvate coming from glycolysis, it's contribute to protein amino acid synthesis. And alpha ketoglutarate generated from TCA cycle contribute to protein synthesis. And the acyl-CoA, acyl-CoA, in the transition step, the lipids contribute to the lipids formation. So these just give you an example. Lots of the precursor metabolites will be generated. It's like an intermediate products used in the anabolism process can be used. So they generate during the catabolism, but they use to synthesize molecular, a new molecular. Basically, it's protein lipids, most of them, and that called the precursor metabolites. Okay, now this is a central metabolic pathway. So we want to talk about these, and then typically we want to talk about these calculations. So the central metabolic pathway, when we talk about here, is actually coming from a glucose. This glucose will go down at a branch, generate pyruvate. This is called glycolysis. And you know that. Then the pyruvate, what they have, it depends where they go. If in the absence of oxygen, without no oxygen, they will do this pathway. They go to fermentation. If they're with oxygen, in the presence of oxygen, they will go to this is called the transition step. They will generate acyl-CoA. And both of these acyl-CoA will go through. This is called the TCA cycle. Okay? Now, we missed one thing here. Something will happen right here in this corner. This is pentose phosphate reaction. This is used to generate the precursor of DNA and RNA, and this is related to NADPH, is the electron carrier, or here rely on FAD or NADH. This is what this slide talks about, central metabolic pathway. So the idea is we oxidize glucose, generate carbon dioxide, and then they use for the biosynthesis. The glycolysis, we'll talk about that. And here is 6 carbon, and become here we go to the 3 carbon. Okay, the pentose phosphate right here is NADH, is an electron carrier or the precursor products. At the end of the day, they generate DNA and RNA. Central TCA cycle right here, which is tricarboxy acid cycle, TCA cycle, or citric acid cycle. They will oxidize pyruvate from the glycolysis. Rely on the transition step with acetyl-CoA. Okay. At the end of the day, of course, it's generate ATP. So later on, we're going to talk about the ATP calculation during the complete aerobic respiration. And we talk about the fermentation a little bit. Then in this class, we will have a specific section later on talk about fermentation, especially the beer brewing and the wine production, including some of the products, because that's related to the fermentation. Lots of the chemicals we can talk about. Okay, this is I already wrote there. Glycolysis, pentose phosphate pathway, a tricarboxylic acid cycle, and generate ATP, reducing power and the precursor metabolites. So we already talked about these. This is basically what I draw here is corresponding to the figure right there. Now, so we're going to talk about this later on. Okay, respiration. There is some of the concepts we're going to talk about. So what is respiration? What is fermentation? Very easy to understand it is using electron transfer concept. What is respiration? So here is an electron donor. Here is an electron acceptor. So first of all, let's talk about what is fermentation. The best definition to define fermentation is electron donor is an organic chemical. An electron acceptor is also an organic